Okay, so problem we're investigating with this lab has to do with the second point of the cell theory. Okay, and that is that cells carry out the basic functions of the organism. All right, so if they're going to do that, and we're looking at eukaryotic cells here, okay, so we're talking about cells from organisms like ourselves, plants, multicellular plants, okay, things like that, then there's got to be certain structures in those cells that do those jobs. Agreed? Okay, so that's the problem we're investigating. Basically, what structures are going to be necessary to carry out the basic functions? Okay, that's the problem we're investigating. So you got to restate that in your own words in the form of a question, same as always. All right, in the design, okay, similar to the last lab, are we looking at the same thing every time, or are we looking at something different every time? Something different every time. We're looking at, okay, a plant cell. We're looking at an animal cell. We're looking at blood cells, although that's only for curiosity's sake. Everyone just always asks to look at blood. Okay, it's not part of the lab. It's not going to be part of the report. It's just there so that you can look at it. Okay, and lastly, you're going to look at a swab of your own cells. Okay, we're going to take a Q-tip and go like all CSI, swab the inside of your cheek, smear it onto a slide, make a wet mount, and look at your own cells. Okay, so basically we're looking at cells from multicellular organisms. All right, but we're looking at cells from different kinds of multicellular organisms. That's probably important because, well, if we want to say that all cells do this thing, we got to look at cells from all different kinds of organisms. Making sense? Okay, now we also said that what are we looking for inside of those cells? Okay, organelles, like specific ones, right? We probably want to say, you know, that it's going to need the following ones. Okay, so we're looking for structures, organelles within the cells. Okay, controlled variables, similar to the microscopy lab, but there's one extra one that I'm going to give you, but you're going to have to explain it. Okay, and I've actually already mentioned it. All the cells we're looking at are from eukaryotic organisms. Okay, so we are only looking at eukaryotic cells. That's cells that have organelles. It would be pointless to do otherwise. Okay, so we won't be looking at bacteria, okay, or other prokaryotic organisms because we won't see any of the structures necessary to carry out the basic functions. Right. All right, for our hypothesis. The problem we're investigating is cells have to carry out the basic functions. What kind of things are they going to have in them? Does that set itself up pretty easily for an if, and then type of hypothesis? Okay, the if part should probably talk about, what do you think? Right, okay, the first part, the if part, should talk about what cells have to do. Okay, and we've said that now, I think, about a half dozen times already. Okay, the and part is always describes the experiment, and the then part should specifically tell me what things you expect to find. Okay, be specific. Don't just say, I'll find the stuff, because I'm not going to be impressed by that, because I just said, don't do that. Okay, so if you do that, I will be enraged. All right, name some specific things that you will find or that you expect to find. Because again, we said yesterday the problem with the microscopy lab is we didn't give ourselves the opportunity to be right or wrong. If we say I'm going to see these things, then we got to be right or wrong. We're either going to see them or we're not. Everyone follow? Okay, so the materials. We're going to change this, not using a toothpick anymore, using a cotton swab instead. I would say Q-tip, except they're the knockoff no-name brand, not Q-tip brand cotton swabs. Okay. Reason we're doing that, I had too many people who insisted on drawing blood with the toothpick. Like they would just scrape it in there and then they'd pull out this bloody gobby mess. Okay, so yeah, not cool, because well, there's all kinds of communicable diseases that can be passed through blood. Okay, saliva, yeah, there's still some gross stuff in that, but it's not likely to contain hepatitis and HIV and things like that. So, okay, we're just using a cotton swab now. It does a very sufficient job of pulling cells off the inside of your cheek. They come off real easy. If it's good enough for CSI and they can do a DNA test, it's going to be good enough for us. All right. So you'll gently swab the inside of your cheek. Proper technique for doing that is to twist the cotton swab as you run it around the inside of your cheek. Okay, so always make sure that you're spinning it at the same time so you get all sides of the cotton swab. All right, and then similarly, when you put it onto the slide to make your wet mount, okay, so basically you're only going to use kind of this central part here, a big enough area to cover with your cover slip. Okay, you'll take the cotton swab, and while spinning it, you'll make kind of a zigzag pattern. Okay, and you're not going to see it on the podcast, but you can see that I'm twisting my hand here as I do this. Okay, you want to twist the cotton swab back and forth. Okay, spinning it in your hand as you go back and forth on the slide. That way you smear as much of your cells onto that slide as possible. Okay, everyone with me there? All right. Then you'll put a drop of water on there, put the cover slip on, same as you did with the letter E. This time you don't have to worry about it flipping upside down or not, just slap it on there. Okay, that's the one you're going to look at last. The reason for that, it's going to be the hardest one for you to see because it's not stained. Okay, all the other cells, all the other, slides you're looking at have had a dye added to them. And as a result, the structures all pick up dye differently and they're very easy to see. In the cheek cell, that's not going to be the case, so you're going to have to make a small adjustment on the microscope in order to see them. Okay, that small adjustment is underneath the stage. Okay, underneath the stage, you see this. There's what's called a condenser. Okay, it looks like kind of a tube that's underneath the stage, and there's a little tab on it. Okay, that opens and closes the iris. You might have used that inadvertently in the microscopy lab. Okay, when you get to the point where you're trying to see the cells, you're going to want to close that off a bit. The less light that comes through increases the contrast of what you're looking at. Okay, and contrast means the difference between light and dark, and that's all you're going to have for an unstained specimen is the difference between dense structures and less dense structures, making them light and dark. All right, so you adjust that, and once you get to the right point, you'll know it because the cell will just jump out. Not literally off the slide, but okay, it'll kind of jump out in appearance. Everyone follow on that? Okay, if you're having trouble with that one, just call me over. I'll give you a little hand with it. All right, the first slide that we're going to look at is going to be a cross-section of a leaf. Okay, that's this picture here. On the slide are leaves from two different kinds of plants, and it's important that you look at the right one. Look at the one that looks like that. Okay, all right, it's going to have a very distinctive top layer on it, made of many rectangular shaped plant cells. You want to draw one of these rectangular plant cells, okay, under high power magnification. That's what you want to make your lab drawing, or sorry, what you want to take your picture of, okay? Everyone follow me there? Okay, if it looks anything other than like this, you're not looking at either the right part of the leaf or you're looking at the wrong leaf. Because what they've done, on your slide, you'll actually be able to see this. The plant cell says leaf, and it says CS on it. It stands for cross section. What that means is they took a leaf, laid it flat like this, and cut it this way, so you're looking on at it from the side. All right. So what you'll see on the slide is this: two little, really fine green triangle-looking things. Okay, and you need to look at the one that looks like this. One is from a monocot, and one is from a dicot, and they're different types of plants, so they have slightly different leaf structure. Okay, so you want to look at the one that has a layout like this. And we're actually going to go over the different parts of this once we start talking about plants a little bit later. Okay, this is the Palisade layer. Okay, that's the layer that you want to be finding a good plant cell on, because it's going to look pretty much like that. Okay, everyone follow there? Okay, second cell you're going to look at is the animal cell. And on the slide, it's going to say stratified squamous epithelium, which is a really technical term for when you dust your knuckles, that red skin that hurts like crazy underneath. Okay, so if you skin yourself, right, and underneath is that red layer of skin underneath, that's your stratified squamous epithelium. It's the underlayer of your skin. Okay, this is what it looks like under the microscope. Okay, looks kind of like when mud dries up and it kind of gets cracks in it. Okay, it kind of looks like that. Each one of these little pink dots is the nucleus of a cell. So the cells are quite small. All right, so if I was, let's maybe do this. Maybe this is smarter. Okay, so if I was looking at this here, that's a cell. Okay, and that's the nucleus of the cell. Okay, those are the kind of things that we're looking for. Right now, if you look closely at this, you can sort of see that there are some darker structures in there. Yours will be a little clearer than this one is. It also looks as though the nucleus has a bit of a beard, okay, on this edge of it. And we said that sometimes there's a certain structure that's attached to the nucleus. Okay, so look at this and look at it discerningly. There are structures you should be able to see, and there are ones you shouldn't be able to see. All right, like you're not going to see ribosomes. Don't label them; they're too small. You'll never see them. All right, but you could probably see things like rough ER. Okay, I would say that this over here, where it's kind of just a light pink shading, that's probably smooth ER. I would say that this little deal right here in the corner is most likely a Golgi apparatus. Okay, things like that. So, I mean, I'm not saying use your imagination, but use your imagination a little bit. Okay, you have to get it as magnified as you can. Look at several other surrounding cells, see if they got similar types of things in them. Okay, maybe one's a little clearer than the other. Okay, but only draw one cell in your diagram. Okay, or if you're taking a picture of it, only label one of them. Okay. All right, so that's what you're looking at for the animal cell. Okay, and like I say, you're probably going to want to find ones that are in an area that's less dense, like this, because the cells will be bigger. Okay, and they'll be easier to see. If you're looking down here, it's going to be really hard. The cells are squished together. They're packed much more densely, and it's going to be really hard to see structures. You'll also see in some places there'll be pieces of scar tissue, or a capillary might be cut. Okay, in a place like that, and you see this kind of big dark red spot. Don't label anything around that. Okay, because it's just the cells are really compressed in that area, and they're really hard to label. So look for cells that are as big and stretched out as possible. Okay, blood's going to look like this. Okay, the thing about blood cells is that you can't see what's inside of them because their membrane is covered in hemoglobin, which is carrying oxygen to the parts of your body, so it's very difficult to see inside of a blood cell. That's why it's not going to be part of our analysis. Okay, so when you're doing the analysis questions, forget about anything you saw with the red blood cell, because you're not going to see anything, and then you're going to have to say, well, I didn't see anything in the red blood cell, so I can't really confirm my hypothesis because, no, just ignore that. Okay, the red blood cell does have all that stuff inside, we just can't see it. All right, so you just look at that, take a picture of it, okay, but it's not part of the analysis. This is what your cheek cells are going to look like. Okay, they'll be spread out quite a bit, hopefully. Okay, but cheek cells are going to be quite light. Okay, this is a picture that I took last semester of mine. Okay, so I had a couple of pretty good ones. Okay, I'd say like this one was the one I've got circled. That one's pretty good. That one's decent. Okay, this is okay. That's a piece of food. Okay, if it's really dark, it's not a cell. Okay, if it's really dark, especially if it's like green and stringy, right? It's something you ate. It doesn't matter how well you brushed your teeth or whether you gargled with Listerine or whatever, okay? You're always going to have some debris that stays in your mouth that's really small. It sticks to your cheeks. It sticks to your teeth. Even if you brush it, it's still going to be there, okay? So don't get embarrassed is basically what I'm saying. Everyone's going to have little pieces of food in this sample. But if it's really big and really dark, it's not a cell, okay? It's probably some piece of fiber from your toast or bagel or whatever it is you had. Everyone follow me there? Okay. If you're not sure, call me over. Okay. But the cells, they look pretty much like the animal cell that we looked at in class. Okay, that we labeled. Right. It's pretty easy to see structures in this one. If I blow it up a little bit here. Okay. I mean, so certainly on all of the cells, you can see the outside, so you should be able to label a what? A cell membrane. Even the plant cell, to an extent, you can see it's going to have a wall, obviously, but you can also see that just inside the wall there are structures. You can see all these little kind of circular things in there. Okay, they're kind of green. Okay. Again, use your imagination when you're looking there, but okay, you should be able to see those kind of really important structures. And here you can see the nucleus, but you can also see that there's quite a bit of stuff surrounding it. I would say that's probably rough ER, stuff like that. Okay, looking for other structures, I would say that we probably have a Golgi right here. Okay, you can almost see that it's kind of V-shaped right there. That little thing that I just obliterated with my blue marker. Okay. Okay, use your judgment. When you're seeing something, use your judgment. If you think that's what it is, say that's what it is. All right, you're probably right if that's what you think. Okay, I would suggest taking your cell diagrams across with you. Okay, so you can kind of compare back and forth. All right, questions there? Okay, so that's what we're looking at kind of procedure-wise. Okay, for the analysis, there's only one analysis question. So how much am I going to be expecting as an answer for this analysis question? Like a pretty explanatory paragraph. Okay, so based on your knowledge of cell structure and your observations of different cells in this lab, what organelles are most important for a cell to function, and which ones are specific to a certain mode of nutrition? Okay, so you're going to have to review kind of your modes of nutrition. We are looking at a plant and an animal here. Okay, and then explain your reasoning. All right, so what I do not want to see is a list of organelles you saw, and then plant had chloroplasts, the end. Okay, I mean, if that's all you're right, expect 1 out of 5. All right, I want an explanation as to why you saw things, maybe why you didn't see some things. Okay, did you expect to see something, didn't see it? Why might you not have seen it? All right, then your conclusion, restate your hypothesis, accept or reject it, and in that explanation, make sure you talk about what you saw. Okay, if you saw all the things that you predicted, then you can say you're right. Okay, if you saw some of them and not others, you can say you're partially right. Okay, but make sure you explain that. And then sources of error, okay, and suggest improvements. Okay, I would say similar to your microscopy lab. Okay, for that one. Questions before we head across to the biolab? Okay, in the biolab front table, all the slides are there. Plant cell, animal cell, blood cell, cheek. Make sure you do it in that order. One slide at your table only. What's that? Plant cell, animal cell, blood, cheek. All right. Okay, the door's unlocked, so you quietly head down there.